Hi, I'm Aurel Enriquez, and this video contains part one of our discussion on chapter 18 of Mahon's textbook of Diagnostic Microbiology. And for this first part, we'll be focusing on the Haemophilus species. Here is the outline of our discussion. Let's first describe the general characteristics of Haemophilus species, their microscopic morphology, habitat, and nutritional requirements, including tests to determine their X and V factor requirements. Next, we'll compare their pathogenesis and modes of transmission, and then we'll determine the appropriate culture media required for the isolation of each Haemophilus species. Let's start with the general characteristics of Haemophilus species. Haemophilus are gram-negative and pleomorphic, meaning that even though we have established the fact that they are bacilli in form, they could still vary in appearance. So hindi po pare-parehas or hindi consistent yung itsura nila. Indirect smears from clinical samples, they could be seen as short cocobacilli, just like these ones in this photo. But if we first take these clinical samples and then make cultures and then isolate this organism, mag-iiba po yung itsura nila. And they will appear like long filaments as shown here. Okay, so ayan po. Haemophilus species are also non-motile facultative anaerobes, and some of them are part of the normal flora of the mucous membranes in humans. So shown here are their other physiologic characteristics. They're also non-motile, so wala po silang kahit anong cell appendage that's going to help them with movement. They are facultative anaerobes, they can ferment carbohydrates, and they're positive for both catalase and oxidase. Listed here are the 8 species that has been associated with humans, but in this discussion, we'll be focusing mostly on these three right here: Haemophilus influenzae, Haemophilus aegyptius, and Haemophilus ducreyi. And of course, we'll talk about these diseases associated to them. Do take note of this. May common names po yung dalawa sa kanila. So Haemophilus influenzae is otherwise known as the Piper's bacillus, and Haemophilus aegyptius is also known as the Cockweek's bacillus. Since we now know that Haemophilus are fastidious organisms, ano po kaya ang mga growth factors na nire-require nila? Their genus name, Haemophilus, was derived from the Greek words haima, meaning blood, and philia, or loving. Hence, the genus name literally translates to blood lover, which makes perfect sense considering that they require nutrients that are present in the blood for them to grow. So there are two growth factors present in the blood. First is the X, or the unknown factor, which we now know as hemin or hematin. So it's not actually unknown anymore. It just has always been referred to as the X factor. Another is the V factor, or the vitamin factor, which is composed of nicotinamide adenine dinucleotide, or NAD. The V factor could only be found within RBCs. Hence, ang V factor po ay hindi magiging accessible sa isang organism if the RBCs on that culture media have not yet been hemolyzed. Now, there are also species that they do not need both of these growth factors. So may mga species po na ang kailangan lang nila X factor lang, or yung iba po V factor lang. And those that need only the V factor have been conveniently named as those with the prefix para. So in this example, we have Haemophilus parainfluenzae, parahemolyticus, and Haemophilus parahemolyticus. All right, so as we can see right here on this photo at the lower left, no, their growth was only observed on the strip that has been labeled V, which contains only the V factor. So hindi po nila kailangan yung X factor for their growth. Both the X and V factors are found within red blood cells, although only the X factor is readily available. So, for some of the Haemophilus species to be able to acquire the V factor, the RBCs in the culture media needs to be hemolyzed. Therefore, they cannot grow on BAP or SBA. Rather, chocolate agar is needed to isolate these organisms. Ulitin po natin, ang X factor po, readily available siya, makukuha siya agad ng mga bacterial organisms if they require the X factor, but the V factor is not. Okay, it could only be found on the inside of the RBCs, which is why clinical laboratories use the chocolate agar for recovery of Haemophilus feces from clinical specimens. Kasi nga po, most of them also require the V factor, which again, you need to first hemolyze the RBCs to be able to make the B factor more accessible to the organisms. In the laboratory, na-discover po nila ang phenomenon of satellitism. So for organisms that need both the X and V factor, ma-observe pa rin naman po natin yung growth nila on blood agar plates. But how? We may be able to observe what is known as satellitism. So as previously mentioned, if this Haemophilus species requires both the X and V factors, they can't grow on SBA, at least not by themselves. So, if, for example, Haemophilus influenzae is streaked right next to a beta hemolytic organism, such as Staphylococcus aureus, that organism then causes the lysis of the RBCs in the medium, releasing the V factor from those RBCs and making them available to the Haemophilus. So, growth will be observed in the immediate surrounding of the Staphylococcus streak. So, again, this phenomenon is known as satellitism. In this given example, ito po, no, by itself, hindi kaya nung Haemophilus influenzae na mag-grow sa SBA or BAP lang. Pero since katabi niya, yung streak ng Staphylococcus aureus na nagpo-produce ng beta-lysine that would hemolyze the RBCs in the media, magiging available na rin po sa kanila yung nicotinamide adenine dinucleotide. Because again, the RBCs have been hemolyzed. So the NAD has been made available in the immediate surrounding. Kaya na-observe na po natin ang growth ng Haemophilus influenzae but only around the streak of the Staphylococcus aureus. So again, that's what we mean when we say satellitism. A lot of the previously mentioned Haemophilus species are normal flora of the upper respiratory tract of healthy people. Non-encapsulated strains of Haemophilus influenzae have been isolated in children, while for adults, about 10% of their normal flora are composed of Haemophilus parainfluenzae, as well as non-encapsulated Haemophilus influenzae. Okay, so ulitin po natin, maraming mga Haemophilus species ang considered as a normal flora of the upper respiratory tract. Let's now proceed with the specific species, and first up is Haemophilus influenzae, commonly known as the Piper's bacillus. Haemophilus influenzae's most significant virulence factor is its capsule. This would remind us of Streptococcus pneumoniae, di ba po? The causative agent of low-burn pneumonia. So parehas lang po sila na may polysaccharide capsule. The polysaccharides that make up this capsule has served as the basis for serologic grouping. And the most invasive infections were attributed to Haemophilus influenzae serotype B. So, in unvaccinated children, this has been a causative agent of meningitis. Keep in mind, though, that not all strains of Haemophilus influenzae are encapsulated, and without their capsule, we cannot serotype them into which serologic group they should belong to, and therefore they are referred to as the non-encapsulated strains that would be or that is known no, that is more known as the non-typable Haemophilus influenzae or the NTHI. Aside from the capsule, Haemophilus influenzae has a few other virulence factors. Immunoglobulin A proteases destroy secretory IgA. So IgA is usually found in the secretions of mucous membranes, providing protection from pathogens along those areas, such as the respiratory epithelium. So it's because of the IgA protease that Haemophilus influenzae can successfully colonize the host's upper immune system. Another virulence factor are their adherence mechanisms, specifically those of the non-typable Haemophilus influenzae. So since NTHIs are adherent to whichever surface they colonize, they tend to be more localized infections. So, for example, acute conjunctivitis. Let's compare that to encapsulated Haemophilus influenzae. So, yung mga yon, they are non-adherent and would most likely cause systemic infections. Lastly, Haemophilus influenzae have outer membrane proteins that paralyze the sweeping motion of the ciliated respiratory epithelium. So in this GIF right here, yan po yung usual or parang visual representation ng usual movement ng ciliated respiratory epithelium natin. But again, outer membrane proteins and lipopolysaccharides have the capability to stop this movement. Here are the list of infections that may be caused by both encapsulated and non encapsulated Haemophilus influenzae. Notice that the encapsulated strains cause a generally systemic disease such as septicemia or arthritis, while the non encapsulated strains with adherence mechanisms will cause localized infections such as conjunctivitis or otitis media. Okay, so ulitin po natin yung mga encapsulated strains katulad ng serotype B, wala silang adherence kasi nga po nababalot sila ng capsule. So they tend to be more systemic with regards to the infections that they cause. But for the non-typable Haemophilus influenzae, they don't have capsules covering them. So they have adherence mechanisms. So the infections that they cause are more localized. Meningitis due to serotype B Haemophilus influenzae used to be very rampant before vaccines against it were available. Here in the Philippines, we have what is known as the pentahib vaccine. So meron naman po tayong bakuna laban dito. So pentahib is a pentavalent vaccine, which means that this is capable of providing protection against 5 different diseases contained in one shot. Okay. So this pentavalent vaccine gives protection against diphtheria, Corynebacterium, or diphtheria, which is caused by Corynebacterium diphtheriae. It could also work against tetanus, which is caused by Clostridium tetani, pertussis, which is caused by Bordetella pertussis, polio by the polio virus, and Haemophilus influenzae type B. Okay, so ulitin po natin, yun po yung meaning when we say pentavalent. Okay, the pentahib vaccine is a pentavalent vaccine, which again could help our immune system protect us against 5 different diseases in one vaccine. Okay, so again, kasama na dito yung Haemophilus influenzae type B. So for this one, symptoms start off with a very non-specific manner. No? So it starts off as a mild respiratory disease with headache and stiff neck before the patient eventually begins to show signs of meningitis. Because, as mentioned earlier, encapsulated Haemophilus influenzae do not have adherence mechanisms, and therefore it is not unusual for them to spread from the upper respiratory tract to the nervous system. Haemophilus influenzae can also cause epiglottitis or the inflammation of the flap of cartilage at the base of the tongue. Epiglottitis has a rapid onset and could cause complete airway obstruction, especially for children since their airways are smaller. So the photo on the right shows us a significant size difference between a normal epiglottis, which is this one right here, versus an inflamed one. So notice, di ba, mas makapal, it's much thicker compared to the normal size. So, ayan po, no. In relation to the next disease that we will be discussing, I would like you to take note of where the trachea is. Okay, so ulitin natin, ito yung epiglottis right here, and then the trachea is the airway which is found right here. Okay, so again, in epiglottitis, there is inflammation and edema of the epiglottis, which could lead to complete airway obstruction. In bacterial tracheitis, the patient accumulates thick secretions that can occlude or block the trachea and is just as fatal as an inflamed epiglottis. So, differentiating with epiglottitis, in this case of bacterial tracheitis, the epiglottis is normal size right here, but the trachea almost does not have much space left for the air to pass through. Okay, so yun po, no, this is more fatal for young children. This is life-threatening for young children kasi po maliit na nga yung airways nila, tapos mas liliit pa dahil ayun nga po, no, there are thick secretions accumulating on the trachea. So again, that is bacterial tracheitis. So just a recap, the diseases associated with Haemophilus influenzae would include meningitis, epiglottitis, and bacterial tracheitis. Now, before we proceed with laboratory diagnosis, let's first look into the other clinically significant Haemophilus species. Next is Haemophilus aegyptius, also known as Cockwake's bacillus. Haemophilus aegyptius was named as such due to obvious reasons. It was first isolated from conjunctivitis exudates of Egyptian patients. So, acute purulent conjunctivitis is more commonly known as pink eye because of how pinkish the small blood vessels in the conjunctiva would appear while they are inflamed, as you can see right here on these photos to the right. So, ayun po, according to the Centers for Disease Control and Prevention, symptoms of pink eye include the pink or reddish discoloration of the whites of the eyes. There's also increased tear production, so nagluluha yung pasyente. Next is feeling like a foreign body is stuck in the eye. So there is itching, irritation, and burning. So parang may something daw po dun sa mata na hindi natatanggal kahit i-rub yung mata. Okay, so the feeling that a foreign body is stuck in the eyes, and there is discharge production consisting of pus and mucus, and also there is crusting of the eyelids or lashes, especially in the morning. So parang nagmumuta ng sobra yung pasyente. Those are the symptoms of pink eye. This next pathogen is actually a different species from Haemophilus aegyptius. This is Haemophilus influenzae biogroup aegyptius. It is the causative agent of Brazilian purpuric fever, and its symptoms include recurrent conjunctivitis. So paulit-ulit daw po yung conjunctivitis, no? So parehas lang din naman yung symptoms na nabanggit kanina: excessive tear production, itching and irritation of the eyes, there is discharge coming out. So ayan po. But there is also high fever and rashes, and this could progress to septicemia and vascular collapse. All right. So as the name implies, no, purpuric fever, may purpura or rashes yung pasyente. All right. So again, the clinical manifestations of the Brazilian purpuric fever include recurrent conjunctivitis plus high fever and petechial or purpural rashes. All right. So again, that's for Haemophilus influenzae biogroup aegyptius. Next is Haemophilus ducreyi. This is not part of the normal flora of humans, but it can only be isolated from humans during the course of infection. Ulitin po natin, no, Haemophilus ducreyi is a strict human pathogen. It's not capable of infecting any other species, but it's also not part of the normal flora. So kapag na-isolate po natin ito, this kind of like confirms that there is a disease that is happening. Okay, so Haemophilus ducreyi is the causative agent of chancroid or soft chancre, not to be confused with hard chancre that is observed in cases of syphilis. Ito naman po is caused by Treponema pallidum. So again, Haemophilus ducreyi causes soft chancre or chancroid. The incubation period ranges from 4 to 14 days upon initial exposure. The disease that it causes is also referred to as GUD or the genital ulcer disease. Now, because chancroid facilitates the transmission of other STDs, all patients who have had GUD should also be tested for human immunodeficiency virus or HIV along with syphilis and herpes virus. This is because the ulcerations that are being produced on the infected area serves as a passage for all the other sexually transmitted infections to enter the host. Kasi po, di ba, our intact skin or undamaged mucous membranes, they're supposed to be a physical barrier that protects us from the entry of pathogens. So, when a person develops genital ulcers because of Haemophilus ducreyi, that's one less defense against other sexually transmitted infections. After an incubation period of approximately 4 to 14 days, a painful lesion with an irregular edge develops generally on the genitalia or perianal areas. The most common signs of infection are on the penis or the labia or within the vagina. Next, pus forming enlarged and draining inguinal lymph nodes, referred to as buboes, are common in most infected patients. All right. So, in this example right here, these are the lesions that are observed on the labia of a female patient. And then these ones right here could be found on the penis of a male patient. Let's now look at two other miscellaneous Haemophilus species. Haemophilus para influenzae. Again, along the first parts of our lecture, what did we say about Haemophilus species with the prefix para? Anong growth factor lang po ang kailangan nila? Just the V factor. Okay. So Haemophilus influenzae can be isolated from the oral cavity of humans. So ito po, pwede siyang ma-isolate as normal flora of the oral cavity. And they are able to cause endocarditis, specifically along the mitral valve of the heart, as you can see right here in this illustration. So from the oral cavity, 1 month after an improperly performed dental procedure, they can migrate to the heart and cause pathogenicity. Next is Haemophilus parahemolyticus. All there is to know is that this can commonly be found on the upper respiratory tract of humans, and it may sometimes cause pharyngitis. So now, what are the methods of lab diagnosis for the different Haemophilus species? Here is a list of possible specimen sources for any of the previously discussed infections by the different Haemophilus species. If the physician suspects that this is a case of meningitis, then the appropriate specimen would be CSF. Bronchial washings are recommended if the disease is respiratory in nature, and so on. Always make sure that the sample to be collected correlates with the clinical manifestations. It's important to remember that Haemophilus species are fastidious, meaning they require additional growth factors for us to successfully recover them from clinical specimens. Most routine culture media do not support the growth of Haemophilus species, so they will not grow on McConkey agar. They will also die rapidly if not transported properly or processed immediately. So minsan po, kapag the sample is suspected to contain Haemophilus, ang ginagawa po is nagbe-bedside inoculation na lang po. When collecting specimens from suspected cases of Haemophilus ducreyi, everything needs to be sterilized because our goal is to isolate the organism causing infection. Therefore, there should be as little to no contaminating organisms if possible. First up, ayan po, no, genital sites first should be cleaned with sterile gauze, moistened with sterile saline. So, ganun po, no? Ganun yung tamang way ng paglilinis ng site before sample collection. Because again, di ba, Haemophilus ducreyi causes a genital ulcer disease. So, before we actually swab those areas, we have to first clean the site para po, ayun nga, ma-minimize sana natin yung pag-collect ng mga contaminating normal flora. Next, a swab pre-moistened with sterile phosphate buffered saline should be used to collect material from the base of the ulcer or yung pinaka gitna at pinakailalim, yung pinakamalalim na part daw po ng ulcer, dun po tayo dapat kukuha ng sample. Okay, so notice that everything mentioned here should be sterile. All right? So, ayun po. Up next, what else do we need to know? The most important thing that you have to keep in mind here is that if collecting swabs for the isolation of Haemophilus ducreyi, we have to swab at the base of the ulcer. Okay, so at the base of the ulcer, because that is where most of the organisms will be found. Direct plating on selective media at the bedside is preferred instead of using a transport media and specimen processing in the laboratory should occur soon after collection for maximum recovery. So the preferred culture media would be the chocolate agar or the Nairobi biplate medium. And listed here are its optimum growth requirements. The best temperature would be at 33 to 37 degrees Celsius. Next is we should incubate this at a capnophilic environment where it is provided with 5 to 10% carbon dioxide, and the duration of incubation is up to 7 days. Alright? So hindi lang sila fastidious, slow growing din po sila. Kasi baka mamaya after 18 to 24 hours, wala pa tayong ma-observe na growth. So we don't automatically report that as negative. So sometimes we may extend the duration of incubation for up to 7 days. Listed here are the contents of the Nairobi biplate medium. Now, the purpose of the vancomycin is to reduce the growth of commensal bacteria from the patient's skin, which we consider as contaminants. While some of the normal flora are inhibited, Haemophilus ducreyi will continue to grow since Haemophilus ducreyi is resistant to vancomycin. So ito po yung parang example or itsura ng Nairobi biplate medium. Kapag sinabi natin na biplate, there are two separate sections in the plate. Okay? So hindi katulad nung usual na full Petri dish na ginagamit natin in our laboratory activities. Ito po may hati sa gitna. Alright? So again, that's what we know as a biplate. Colony morphology of Haemophilus ducreyi appears to be small, flat, non-mucoid, tan to yellow colored, and if you take a clean inoculating loop and try to push one of the isolated colonies to the side, they will move but remain intact. So in this GIF right here, this is actually an example of Moraxella catarrhalis. Kaya po nakikita nyo na BAP yung culture media, di ba? Pero ulitin natin, Haemophilus ducreyi would not grow on BAP. But ito po yung sinasabi natin, no, na colonies could be pushed across the agar using a loop. So, magamit po yung inoculating loop, imbis na sasama basta-basta yung isang colony, they stay intact and then they just move together. Alright? So, kaya din daw po itong gawin ng Haemophilus ducreyi. But again, in this GIF, ang pinapakita po ay Moraxella catarrhalis. I just added the GIF para maintindihan nyo po yung sinasabing motion. For Haemophilus influenzae, the recommended culture media is the horse blood bacitracin agar. Although for most laboratories, we don't have that, so chocolate agar infused with bacitracin may also be used. They must be incubated under capnophilic conditions. And for Haemophilus aegyptius naman po, an enriched chocolate agar could be used that is supplemented with 1% Isovitalex. Now, if you would remember from our discussion on the Neisseria species, diba, the Thyromartin agar is also supplemented with Isovitalex. It's a commercially available formulation that serves as a nutrient source. So, yun naman po yung preferred for Haemophilus aegyptius. So, ulitin po natin, for Haemophilus influenzae, we should have the horse blood bacitracin agar. For Haemophilus aegyptius, we should have the chocolate agar with Isovitalex. The appearance of Haemophilus influenzae on chocolate agar is described as non-hemolytic, tan, convex, with a mousy or bleach-like odor, which is very characteristic of Haemophilus influenzae. Kapag po nakita niyo yung description na mousy or bleach-like odor, dapat ang maisip niyo po agad Haemophilus influenzae. So imagine a place that is infested with rats. Ganun daw po yung amoy ng colonies nila. Pag sinabi naman bleach-like odor, it's not the clean smell of bleach, but rather if you accidentally inhale large amounts of concentrated bleach, yung masakit na feeling sa ilong, parang ganun daw po yung odor that is produced by Haemophilus influenzae. If the Haemophilus influenzae strain that has been isolated is encapsulated, expect that the colonies would appear larger and more mucoid in comparison to the non-typable Haemophilus influenzae. So an example shown here is ito po, no? So this is an example of an encapsulated Haemophilus influenzae. Haemophilus parainfluenzae colonies will appear dry and slightly larger in comparison to Haemophilus influenzae colonies. Meanwhile, the colonies of Haemophilus parahemolyticus may resemble parainfluenzae, but do take note that parahemolyticus is beta hemolytic. Okay, so madali lang tandaan, di ba? Parahemolyticus, beta hemolytic. At ulitin po natin kapag may para, sa umpisa ng pangalan nila, what growth factor do they need? Just the V factor. Looking at the microscopic morphology of encapsulated Haemophilus influenzae, we may be able to observe the appearance of halos, which are non-staining areas that surround each cell, as we can see right here on this photo. Okay, so again, those halos or yung parang something sa paligid ng bawat isang bacilli, those are their capsules. When it comes to the microscopic morphology of Haemophilus ducreyi, there are two characteristic descriptions that may be observed. First are railroad tracks, like this one right here. So basically, yung mga bacilli or yung mga cells daw po, magkakaduktong sila or magkakasunod sila. Okay? So parang pila-pila sila, no? So when we say railroad tracks, mukha daw po silang riles ng tren. Okay? Next, we also have the description school of fish. All right. So when we say school of fish, they don't have any specific arrangement. Rather, they're just clustered together like this one. We could check their X and V factor requirement. The usual approach to this is either by placing strips that contain the X factor and another strip with the V factor on the agar plate. If growth is observed only around the X strip, it means that the organism only requires the X factor. If the organism grows around only on the side of the V strip, then it only requires nicotinamide adenine dinucleotide. Okay? But if the colonies are seen to have grown right in the middle of the two strips, then it means that this Haemophilus species requires both the X and V factor. Discs may also be used, and in this example, there are separate discs for both the X factor, V factor, and then there's this one disc that contains both of those X and V factors. In more advanced and highly funded laboratories, they have what is known as the Haemophilus quad plate. And as the name implies, there are four zones to streak the bacterial isolate on. So there is an agar base that is infused with either the X factor only, or the V factor only, or both the X and V factor included right here. And then there's also one quadrant that has horse RBCs. So on this quadrant that has the horse blood cells, the organism's ability to produce hemolysis could also be observed. On this table, we see both the clinically significant and miscellaneous Haemophilus species, alongside with the growth factors that they require. Haemophilus influenzae, Aegyptius, influenzae biogroup Aegyptius, and Haemolyticus all require both the X and V factors, while Haemophilus ducreyi only requires the X factor. Now, again, what is the component of the X factor? It's hemin or hematin. Okay, hemin or hematin. Notice the species with the prefix para. Okay, so they only require the V factor, such as Haemophilus parahemolyticus and parainfluenzae. Now, what is the component of the V factor? It's NAD or nicotinamide adenine dinucleotide. Another test that can be done for us to identify Haemophilus species is by the Porphyrin test. The principle of the test is that if the organism can convert delta aminolevulinic acid into porphyrins or porphobilinogen, then they are not X factor dependent. Kasi kaya nilang gumawa ng sarili nilang X factor given that they are supplied with ALA. So these substances are detected by adding paradimethylaminobenzaldehyde, commonly known as the COVAX reagent. And then an ultraviolet light is set at a wavelength of 360 nanometers. And this is used to visualize this reaction. So this UV light is also known as the Woods lamp. As shown in this picture, the organism on the top is porphyrin negative, and the streak at the bottom is porphyrin positive, or it's positive for porphobilinogen production. So this would indicate that the organism on top would still require the X factor kasi hindi niya kayang gawin yung mga precursor molecules, while the one at the bottom does not need the X factor. So let's just re explain that. Species that are porphyrin negative cannot synthesize heme, even if you supply them with delta aminolevulinic acid. And therefore, they rely on being provided with X factor for them to grow. Meanwhile, species that can synthesize their intermediates for the X factor does not need it anymore, and they would only require supplementation with the V factor. So, in summary, if the organism is X factor dependent, it will be porphyrin test negative. But if the bacterial isolate requires only the V factor, then it will be porphyrin positive. Here are the references that were used in the making of this discussion. That ends our discussion on part 1 of chapter 18 of Mahon's textbook of Diagnostic Microbiology, where we focused on the Haemophilus species. This has been Aurel Enriquez. Thank you so much for watching.